All right, now, talking blues, people might just say, well, that's talking blues. We're going to talk about blues tonight, and that makes perfect sense to people who study butterflies. But my brothers down where I work, they got posted on the lunchroom bulletin board that I was going to do this thing. And of course, these guys were amused by the whole notion that somebody that looked like this could do something like butterflies. And so I get no end of grief, and one of my coworkers knows that well enough. So one day I come into the break room, and there's a guy kind of looking at the wall, looking at this thing, and looking at me, and then looking at the wall, and scratching his elbow with his hand. Hey, JP, I see you're giving a presentation. And I said, yeah. He said, well, are we talking blues? And I was tempted at that time to digress and go into the history of the buckwheat blues and evolutionary dynamics of post-glacial dispersal and adaptation, specialization, differentiation, and I said, nah. No? I said, no, it's just a kind of butterfly. It's a blue butterfly. This could be, there we go. Yeah, that's a blue butterfly too. A lot of people that just,
No, it's
it's a blue butterfly.
yeah, very funny.
All right. But this is what, people that think of butterflies that don't know butterflies might say, well, maybe he's going to talk about these. And
the counter that's missing the apple.
no, it doesn't count. And this is what we're going to talk about tonight. Everybody see that?
That's a copper.
Yeah, that's actually relative size difference between these two butterflies. And this is a closer up vision. And we're going to talk about blue butterflies that are more like this one. And they're not less spectacular, they're just smaller. Okay? So to really get admiration, pygmy blue.
Pygmy.
Yeah, we'll get to that because it is one of our butterflies. We're going to kind of look at how these things break down. Now this is half or half the tree from the tree of life of Lepidoptera, and this is the half that really has some meaning to us because the other half involves all these things that are leftovers from the Cretaceous, and they live in southern beech forests, or they're like mandibulate moths, moths with mandibles, so they don't really count even though they're kind of like the major part of the whole stem. But you can see right up there at the top are the butterflies, and I don't want anyone to think that because they're up here somehow they have priority, because really they could be anywhere and they could reverse those trees around and they could be in the middle. It's important to recognize that butterflies really are only specialized day-flying moths. So, those of the people who, those people who, want to think of butterflies as special and people that study them as special, now you just glorified moth collectors or observers. This is a closer look at the real butterflies, all right? They excluded the not real butterflies, which are the skippers, okay? They say true butterflies, but they don't say, the false butterflies. But if these are true butterflies, then these must be false ones, right? I don't know.
It's a fidelity thing.
You don't want to know, man. Why do you ask questions like that? It's the moth-like butterfly, or it's the butterfly-like moth?
Anyone that this might be familiar with?
No, they're all tropical. One might get into Texas, but you don't want to go there, really. Okay, this is the whole arrangement of butterfly families. Now, there's only 5 of them. And some people might say, really, there's only 4 because you can combine these two. But Lycanidae are the ones that include the blues that we're going to be talking about tonight. And so, I just expanded that branch, and you can see that, there's all kinds of subfamilies in that group. And it's really interesting, actually. These 3 here are the ones that we think are important because in the north, where science has, scientific methodology basically evolved, this is where, these butterflies occur. These other things are African or Australasian. And so as a result, we didn't grow up in those environments where, we had a diversity of lichenids flying around. We just had these 3, so we think that's really what lichenids are about. But the Polyomytini are the blues, all right? You got a question? No, you don't.
Butterflies really carnivorous.
Yeah, some of them, the larvae, not the adults. I like more immediate response of my accelerator here. Okay, now this is a breakdown even further, and that's just about as far as I want to go with this. Yeah. Now, need to get through the life history a little bit because it's real important with lichenids. It's real important with blues. Basically, you saw earlier tonight that you had the egg and you have the larval stage. The eggs are really interesting with lichenids. They have this microstructure, and if you go deep inside there, you see some incredible structure. You know, we talked one time, we had a thing on the scales of butterflies, and you saw the infrastructure. Well, it's really fascinating in and around. This is where the sperm enters the egg. So when you think about, you know, the importance of procreativity, the sperm-egg interface is critical evolutionarily, and it probably has significance at several different levels. But hey, you know, that's for another time. Come on, baby. There we go. Now, blue larvae are all of a kind, kind of slug-like in shape. Now, I want to point something out. I don't want to go into great detail about the larvae. But see these little bumpy-like things right here? And there's a total, actually, two on this side and two on the other side that you can't see. Now, these are actually tubercles that have an inverted sac, which, upon stimulation, everts and gives a substance. I don't know what the substance is, but it drives ants crazy. And the reason I'm bringing this up is twofold. One is because blue larvae are going to be associated with ants a lot, and that's one way to find them if you've got nothing else to do. The other thing is that its name is given. It's called Newcomer's organ. And Ervil J. Newcomer was one of my mentors, and he was the one that basically defined and described this. And so we're going to kind of, helps if I press the right one. But these things are pretty bizarre sometimes, depending on what part of the plant they feed on, etc. And then this is a pupa. You saw the really glorious pupa of the American lady. These tend to be kind of like, resembling a rodent drop. Now here we go. This is where we're talking about. This is a real important feature in the blue butterfly biology. Okay, this is a larva being tended by ants, and we have several photographs. We might as well just move right through them. This tending is all so that these ants can get the substance, which is affectionately called honeydew. Now that means basically that anyone using that term says, we don't really know what this stuff is, but ants love it, and honeydew seems to know. It's actually more often associated with aphids. Yes?
Nomi Pearson, her work in Australia showed in that case that it was cocktails of amino acids, fairly basic stuff that's really good for them.
Obviously so. I think there's something fundamentally useful to these ants, but it's also something really attractive. Like it makes them high or something. Because these ants, you just rarely find an unattended, like handed larva in the wild. And I'm just going to go through a number of these. Real attentive. Now, in the most advanced cases of this relationship between blues and ant larvae, the ants will actually carry young blue larvae into their nests, feed them their brood, and to maturity. In other words, they become carnivorous. They eat ant brood. And then upon emergence, they have an elaborate way of escaping ant predation. It's just like, wow, man, this must be an important thing. And I'm going to tell you why a little bit later on. But one thing I want to say about it is that because of this relationship, we have an interesting thing happening in the plants that these blue butterflies use. Because they'll use a plant, one specialist on a certain plant will come in and have this ant association, and then another kind of wander in, and they'll use the same genus of plants. Like say we're talking buckwheats, all right? And so it'll lay its eggs on any number of buckwheats in that environment, but because there's this already extant plant or ant association, an association with this one plant being used by the one butterfly, then the increase of the ant protection that comes as a product of that already existing relationship increases the survival of those that lay eggs on that plant. So you get two. And then, of course, then you've got, like, the ants are like, oh, this is cool. I got Holsteins and Guernseys, And then you go to the extent that 3 and 4 and as many as 5 different species get compounded on one plant. Now, it's true that that plant has to be, a pretty nice plant. We'll talk about some of them that we'll see in a minute. These are juicy, luxuriant plants, and so they have to be able to withstand drought, etc. But once that plant is found, then this guild building of Myrmecophilus lichenids, which would be hoppers, hairspinks, and blues, is a real interesting phenomenon. Okay, some other things. I'm pushing on the accelerator, getting no answer. Do I need to point it down here, Dave, or?
Point it at the projector.
All right. Okay, this is more of the same. Am I going backwards? No, okay. Another thing you're going to hear us say about like blues and lichenids in general is we're going to talk about genitalia. In fact, you might think butterfly people are a little perverted because we talk about genitalia all the time, and we show pictures of mating butterflies. Well, the genitalia are important because this is a structural character that we can actually help to determine specific differences between some of these very, very complicated butterflies. And I'll show you some examples later on, but I wanted to kind of go through some of the terminology. This is the equivalent of the penis. This is a ring that goes around the tip of the abdomen. The penis is inserted in the middle. There's kind of this big blocky thing on top called the tegmen, which is also the uncus. And this is a side view of the same thing. So this here is equal to this here. And then the valves, which they're not really grabbing things. They're more like guiding things in terms of positioning female abdomens. Now, Lars studies moths that have some really fantastic stuff that comes out of the end of the ediegus. They have these elongated membranous things. Butterflies tend not to, although skippers have some more. But we're just going to kind of go through. This is what it looks like when it's broke down into its component parts. It looks real tidy and everything, and that's what it looks like actually on the slide. That's actually a female genitalia, but you can see it's like all of a sudden, while the real world comes to the artist's rendition, and things kind of dissolve. Another thing about blues is that they're on mud all the time. That's why it was part of the title. Why are they coming to mud? Well, essentially there's some minerals, sodium and other sodium and potassium salts that are available as free ions in mud, and it's mostly male, so it seems to be something that's important for their development to maybe sexual maturity. But you're going to see a lot of blues on mud, and one of my favorite stories is about Zach. He was 5 years old up on Chumstick Mountain, and we came to Swakane Springs, where the mud's alongside the road, crosses the road, runs down the ditch. And anybody knows what, July 4th, it's 85, 90 degrees, and all the blues are on that mud like crazy. You walk through there, there's blues just flying up. And, Zach's like, whoa. And he goes, Dad, there's blueies everywhere. Sorry. Now, the other thing that's interesting about blues is that really it's only half right. Now, this is a dimorphic sexual pair. This is actually Celestrina lucia, the Lucian azure. And what you can see here is there's quite a difference between the sexes. In this one, at least the female is essentially blue. But in many, the females are actually not blue, or they have only a scattering blue scale, so they're sexually dimorphic. And that means to most people that, well, really, you should be calling them half blues. What? Okay. Now we're going to go on, and we're going from order of ease of differentiation. Everyone's going to be able to recognize these butterflies pretty quickly. This actually is a Washington butterfly now, although by just the hair of its teeth. Okay, it gets into Washington just barely, and it's a spectacular butterfly, despite the fact that it's often no more than like 5 millimeters across. Now, one of my favorite stories about this butterfly was in California, where we were in a place where there was a lot of this was tumbleweed, Russian thistle, and I think the scientific name is South Sola cali. Well, tumbleweeds are singularly unattractive and pestiferous kind of plant, but in fact, when it's growing, it's wonderful. It's lush and it's very, very vibrant little tree-like plant. It has a lot of substance to it. And as you were walking through these waste areas, and there were these tumbleweeds, live tumbleweeds, there were, there were so many of these things that if you stood at a distance, you couldn't differentiate them individually. There were clouds. And when the slight breeze came up, the clouds would sweep behind in the lee side of these little bushes. Yeah.
John, what was the location?
That was just outside of Bakersfield.
Huh.
Yeah.
And you say it just barely gets into Washington?
We have a total of 2 records
of
which occurs here.
So it doesn't really.
Hey, climate change, there's always hope. We might lose some, but we'll get some more, Yeah, no, it just barely. But see, it's a butterfly that might be able to adapt to regions more close to, and then get here as a product of their adaptation to those close.
So what about Oregon? Is that?
Yeah, it's widely spread in eastern Oregon.
Oh, okay.
But it's not really resident anywhere in eastern Oregon. So it's just a matter of how close the resident populations are. Climate change is affording us an opportunity to possibly see it. I think this is a nice picture. Isn't that a cool picture? Yeah. It's a female. And I don't know.
Some scale there.
Huh?
It's something for scale.
Yeah, I know, that's the problem with BOA images, but how about, you just take my word for it, tiny?
I've seen them. I
Yeah,
thought,
right.
what's this little tiny thing?
Look
It
at,
looks like
look at
it,
the
but-
fingernail on your little finger, that's the size they are.
And they're really terrible to have to mount. I've decided that after a Bakersfield trip that I was gonna put them all on like minute and natal and double mount them on a block of fungus, you know, because that was just trying to stick a pin and putting them in trays was just
What's
impossible.
that food plant?
They have a whole bunch, just listed.
I know, but I don't.
Salsola coli, is they got a different name now?
I think the name's the same, but I think the whole family. It doesn't use a bunch of that family.
Chenopodiaceous.
Chenopodiaceous,
Yeah.
the goosefoot family.
Yeah, goosefoot families are big on.
Okay.
And of course, their ability to use weeds is, you know, really useful. You know, this is a butterfly that actually has been recorded one time from Washington. It's almost certainly an artificial import. But, you know, I'm going through it just, okay, this is what I was saying. This is the other thing that you see is a lot of mating pairs, and people say, what are you guys, prurient? You know, and I say, no, it's really simple. It's like when butterflies are mating, they're quiescent. You photograph them more easily. So you have a lot of people taking advantage of the opportunity to get really spectacular photographs. Now, these butterflies, you're not going to have a problem identifying these. So, you know, really moving on, I'm going to the things, you know, that are really difficult and more interesting. Now, this is what's called the silvery blue. Okay, everybody knows this butterfly because it's like our trip to Shelby Coulee every year. You see it in a lot of places all the way up to alpine levels and Slate Peak. And then silvery blue, you can kind of see why it's called the silvery blue. But the characteristic that identifies silvery blues immediately is that you got this row of spots, both on the forewing and the hind wing, and then there's nothing but vacant space between those spots and the margin of the wing. And that's like characteristic. It wouldn't work in Central Asia or Europe, because there's a bunch of species like that, but it works really well here. And so it's a piece of cake, you know, to figure these out. And, you know, like the figures that we'll see in a minute of live specimens will confirm that, you know, in mine. Now, I do like the idea of having mounted specimens and, you know, like in a rather more sterile kind of frame, especially if you can, you know, be so fortunate as to move on to, you know, live specimens and see, you know, what they look like when they're actually in nature. So the combination together is an astounding feature. Like
There's
butterfly
a female on top of that last one,
Yeah. Which one now?
the mating pair.
No, female's on the bottom there. No, you're right. No, the female's on top. Yeah. Now this is, again, mudding. This is what they do, is set it to mud. And, you know, it's really useful to bring butterflies together. But when we get to the buckwheat blues, we'll find out how bad that is. Because one of the ways you can understand and know buckwheat blues is by seeing them in association with their food plants. It's like, helps you get an idea of what you're actually seeing. Well, if males leave the food plant, go down to the mud, you can have, you know, maybe as many as three different buckwheat blues on the mud, and then, you know, you're kind of at sea. But in this case, it doesn't make any difference. So this kind of gives you an idea of the silvery blue aspect. And again, you can see that the same character that we're looking at here, you know. So this is a piece of cake. This is another one that's a piece of cake, you know. Up on the top side, you know, there's really not much, you know, but this, it's called the arrowhead blue, and these are because these marks appear to be like arrowhead shaped, okay? And remember, guys, I didn't give them these names. Somebody else did that. I did make up some, and we'll see later on, and you guys can vote on whether or not you find that acceptable. But nonetheless, I mean, this butterfly, if you see it in the field, it's not going to be a problem. You're going to be able to identify it very, very easily. And it, you know, that we're really talking about the things that are hard to identify. Last summer, people complained about all the things that have happened with certain groups of blues. Well, these are the ones that didn't happen to, right? Okay. Again, this one here, a lot of times it's easy to figure out just on the basis of where it occurs. It's essentially alpine or subalpine, and generally speaking, it's very alpine, often in really desolate kinds of habitats. And we're going through a couple of different images. Some of these are from the Strickland Museum, and some of these are from the BOA site. Basically, you're going to see a lot of variation, even in a single population, so I don't feel too bad about getting them from different places. There's a certain aspect to the butterfly dorsally that makes it easy to identify if you've got some familiarity already, but in fact, it's the underside that really kind of gives it away. The dark, and well, you know what,
Is the host on that, you know?
There's several, but most of the time it's a saxifrage. And Slate Peak, for instance, it seems highly likely. Well, we've got larvae on saxifrage and rocky owls. But in the Arctic, it's saxifraga, tricuspidata, and you know, it kind of varies. But there's androsace that's been used, and I think there might even be some legume feeding. These things aren't as restricted as people formerly thought. And this is another one that's pretty easy to figure out. They call it the greenish blue. I've never quite figured that out. There's really not much greenish about it. But, you know, it's got an aspect, these spots along the hind wing margin with that little orange thing there, the fact that they're paired off. You know, really people were able to identify this pretty quickly on field trips we've been on, and it hasn't changed too much. You know, people have said, well, you know, that's the same greenish blue on the field trip in 2008 as it was on the field trip in 2003. So, you know, it's really apparently people are catching on to, you know, what this butterfly is, and it hasn't been too much trouble to identify. It's you know, it's a cool butterfly, extremely dimorphic. The females can be so dramatically different from the males that it's almost shocking. But you can be in places like, you know, there's a case in Point, you can see a great dramatic difference between male and female. One of the trips we take up to Lion Rock, there's a meadow along the way there that is so full of these things. It's amazing. And they feed on a clover, which is, I think I have an image of here, which is one of my favorites. That's an egg. Yeah, there it is. And I mean, this meadow's just full of these clovers. And we did a 4th of July butterfly count, I think, for that trip. And Kiyoshi and I decided to walk out and do hectare by hectare, or square by 10 meter square, and estimate how many there were. I think we came up with like 250,000, you know, and we turned in that results in NABA. And I'm sure that just pleased Jeffrey Glassberg no end. This one is a little more difficult, but nothing's really changed. It's the same waterfalls blue that it always has been. This is an easy one to kind of figure out because it's got this halo of you know, around the spots in the median band. It's not silvery blue because you can see all these other markings between the middle band and the edge of the wings. And the other thing is that most all the variation you're going to see involves an increase of this halo to the extent that these spots disappear, and the lightening of the ground color to the extent that even the spots themselves, the halos with the spots disappears. And that's just part of like what we're going to see. Now, this side here, you know, this is a familiar sight to anyone that goes to the east slope of the Cascades in June and July. You're going to see something that looks like this. You say, wow, man, this is the picture that JP showed us last February, when it, you know, it's almost the same that you can see repeated time and time again. Now this is an example of an individual where the ground color is paled out and the spots that are less contrasting. You take that to an extreme and you can see like they've almost disappeared. Now we have in the Cascades what I call the white witches. Back east we have what they call the 3 black witches. Now Lars, do you remember which those were? I can't remember. They're Hesperian skippers. I can't remember. It was Lerodia, Euphila, and 3 Hesperian skippers that are basically dark, with no maculation, and the only way you can really tell them apart is to look really closely. Well, these are very similar. This is one of them. It's the waterfall's blue. The other is western tail blue, which gets really pale. And then the third one's Anna's blue, which can be suppressed. And they all look like white blues, and they'll be the white ventral hind wings on the blue. And they'll all be flying in the same places, so we call them the 3 white witches. And it takes close scrutiny to kind of tell them apart. But now we're going to get to the white form of the western tail blue. But this one is one that very few people get to see in this state, and that's because it actually occurs in a part of the state we go to relatively infrequently. And it's kind of like, actually, the western tail blue, which we do see on a lot of field trips. The main differences are the intensity of the orange spots like here. I mean, they all have tails, they're all the same. And the basically intensity of the spot pattern, period. Now we'll kind of rotate through and see some of those. And you know, as you go into the next. Also, the fact that there's usually a dorsal orange spot on the female surface, and there's usually not in the western. But I'm going to kind of go through in a hurry, and you see, well, you know, some males have that orange as well. You won't ever see that in the western tail blue. And we'll go right to the western tail blue, so you can kind of see what I'm talking about. You know, no orange on the top side, and the orange on the underside is kind of like very restricted. The overall spot pattern is subdued. And in fact, in some cases, it's extremely subdued. Now, he's missing an abdomen. That means somebody was doing some genitalia, and those are things that people box. And this is really suppressed. You can start seeing where we get to the white witch thing. Almost always, though, if you look closely, there's going to be at least a hint or an indication of these orange lunules, and maybe even a little bit of the metallic, so you have to look really close. But, you know, when they're all flying together, sometimes, you know, all it looks like is a bunch of little white butterflies, or butterflies with white undersides. Yep. We'll see a lot of them look like this on the east side. And again, the tail almost always gives it away. The only thing that would ever be a problem is it is eastern or western. And then if you're not in the appropriate places, which is like in the northeast of Washington, you're not really going to see an eastern anyway. So, you know, if you know you're in a place where they could occur, then you have to, you know, go a bit further. But again, this hasn't been a problem. People haven't come back to me and said, dang, man, I can't tell the difference between those eastern and westerns. They're just killing me. You know, most of the time they say, what the heck are these azures that you're talking about? We'll get to them. I don't know what that is. It looks like a mistake. Yeah, we do get some in Washington that are better marked. Okay, now, see, I mentioned azures, and just like magic, it happens, you know. Who's in charge? Huh? Why do you think that happened? All right, Echo azure. Now, hey, 7 years ago, 8 years ago, we didn't have an Echo azure. We called it Celestine Argiolus Echo. All right, and it was like a totally different thing. Well, things have changed, and the only thing that hasn't changed is the butterfly that we all know. It's basically still the Echo blue that other people have recognized, and all the characters by which it can be distinguished. You know, these spots look like spots of eyebrows along the margin of the wings. You know, same butterfly. You know, really is essentially the same butterfly. The only thing is how we recognize it and the name we call it. All right, and oops. Sometimes this does really well, so well, in fact, I get ahead of myself. Now, come on, everyone knows this butterfly. No one's going to misidentify that thing. Notice the color here, okay? This is going to be critical later on. This is I don't know, people have described it as kind of a violet blue. I'm not sure I even know what to describe it as. It's got some elements of luster to it. You know, it's not just a regular blue like a water ball's blue. It's got maybe a little bit of iridescence, and somebody could look at those scales and probably, you know, in fact, haven't you done something like that?
Yep.
Yeah.
You can do some more too.
Yeah.
If they ever come out.
Yeah, yeah. And there was an issue with that last year, wasn't there?
Yep.
So we're moving right along. Now this is the one that people get a little bit squirrely with, okay? And we get these along the east slope of the Cascades in like Kittitas County and in Yakima County, but it predominates in the Blue Mountains. So you guys went to the conference last year and you saw what you thought were Echo Blues over there. They were all of this thing here. And this is, we don't really know what's going on here. We're not sure of what's going on here, but again, a basic aspect is pretty much the same. See how much darker these spots are here? That's just an average individual of kind of a normal form. Sometimes you get these really blocky things, you know, that are really, and even more extreme than this. I looked for the most extreme that I could find, and I went back to Andy and said, we got to get some more of these images of the really extreme forms, because they can be almost where the whole basal area is full of this dark scaling. Certainly not like our Echo Blues, yes.
John, are these multiple brooded? Do you know?
Nigrescens, it's hard to tell. They have sort of a long single brood.
Do they
Yeah,
come
I
out
don't think.
early?
Huh?
Do they start early?
No, not as early as Lucia's. No, they're about like with Echoes. I mean, you know, Echoes are pretty early blue too.
Yeah.
But I put a lot of images in here so you can see that there's really a lot of difference in terms of like the expression. In fact, it's characterized by not being standard. You can go into a place and see lots of these blues, and every one you look at would be slightly different, and it's, you know, kind of a bizarre thing. People have speculated about, you know, where it came from, and we'll get to that when we get to that species, which is just around the bend. And I'm just going, I kind of picked this as much as I could that would give us. Now see, here's something that looks kind of normal, but it's from a population that included all those other whacked out things, and that's part of the deal. You know, is that you get things that look like a pretty typical Echo Blue, and you get this other stuff. But now there's the one that we think may explain what's actually happened, okay? In the course of non-human events before the age of time, there were things that happened. Like glaciers came and they went, and things happened such that populations came and they went, and then one another butted one another at some time. There was some maybe hybridization going on, and that hybridization may have led to the introgression of genes from this species into the echo blue, and that's what we're seeing. Because this species, now you see right off the bat, see how this is a much, much lighter blue. It's very, very silvery color. Anyone who's been to, what's that, Cowiche Canyon, and seen these things flying on our field trips over there, immediately can see, wow, this is really different. It's a much more whitish blue, and the intensity of the dark markings on the underside is severe. I mean, sometimes much worse than that, but never less than that, or at least rarely less than that. No, that's the last one. Got JP some slack here. And, you know, it can be pretty remarkable. And that's, well, this is from Cowiche Canyon, okay? And there's some people that were really surprised to hear that we had these populations in Cowiche. People, you know, considered that this is a butterfly that was pretty much limited to transboreal Canada, from, you know, Newfoundland to Alaska, and penetrated south into British Columbia. And, you know, I didn't have the heart to tell them it's worse than that, friend. It's not just penetrating from this transboreal force, it's going out into a desert canyon. And that's what Cowiche Canyon is. I mean, it probably flies in some of the same places Aberdeen and Wormhole would later in the summer. So it's pretty striking. Now, we haven't resolved all this, but you know, I think most people can identify the taxa. And this is the one we run into problems with. Now, there's two butterflies that have this basic setup, a little blue in the male, showing orange lunules on the hindwing like this, okay? You see this is kind of a pinky orange color, and there's really no other butterfly in the Northwest between this one and the lupin blue, all right? So right away you can, you know, identify down to those two, but the problem is that for a long time we considered that this was the butterfly that existed in Washington primarily, and didn't realize that we had lupin blues at all. And then we thought we were all lupin blues, and didn't realize that we had this one. When you see the list coming out, we'll talk about the fact that this definitely occurs in the Columbia River Gorge, probably occurs much more widely than we ever knew in southwestern Washington. And the reason is we're not looking for it in the right places. It flies in like garbage dumps. I mean, it's really, you're going alongside of the road and you see old cars and, you know, a couch, you know, thrown out by this. That's where you got to stop, because that's where you're going to find these things. They're in really trashy places. They feed on plants that, like lotus, funiculatus, and other things that you know, would be found on the dry side that are really kind of weedy plants. And that's what they feed on. They feed on fabaceous pea family plants. They don't feed on buckwheats at all. So, you know, this is a butterfly you all need to go out and look for, you know much more assiduously.
And this is Ackman, right?
Ackman blue, right.
Double check here. You forgot.
And now, we're going to get to the characters that distinguish it essentially between that and the lupin blue. Now see, where the orange band is on the hind wing, there's almost, you know, there's some spots, there's like an orange halo, maybe even a band formed from a halo of spots, but there's no dark coloring basal to that, in other words, towards the base of the wing. In a minute, I'll have it compared again here. See how there's just this abrupt end between the orange and the blue, all right? And again, I wanted to get as many of these images so that, you know, you guys can see I wasn't just making this stuff up. And the lupin blue, on the other hand, is really quite different. You see how there's this really strong, dark line. It can be less of a line, more like another halo, but it's a definite contrast between the orange and the base color, which is the blue. And that's a female, which is more blue than you would most males. Now, again, here's one where it's a little bit less clear that there's dark color along that orange margin. But you can also see that it's, you know, it's got a kind of a different aspect of color, but it's pretty, you know, pretty suggestive.
Architect sense.
Yeah, don't pay attention to that.
You know,
the man behind the
confuses
curtain,
me.
you know. And I think I was using this, I don't know what I picked that, because that looks like an Ackman. Or maybe I picked it because it looks like an Ackman, but in fact, with the heavy borders here, it's probably just a lupin blue. It's very tough. One thing about it, these all feed on buckwheats, and they only have a single generation a year, pretty much, sort of, maybe. You know, as we're going on, we're finding out that even that's not true. So on the east slope of the Cascades, year after May and June, you're seeing lupin blues because they're feeding on buckwheats. Don't ask me why they're called Lupin Blues. I just can't explain it. But bottom line, Acmons you're only going to find out in the Columbia Basin, and you're more likely to see them in September, August and September along the Columbia River Gorge and whatnot. And when you see them, honestly, when you see them for the first time, like when I first saw them in Kern County in 1965, and when I was there most recently, saw them again, I was like, wow, they really look different from Lupin. They're tiny for one thing, and they look different from Lupin Blues. But we'll keep on moving along here because, yeah, some of these images are really astounding. I really like that one in particular. Okay, now we got a comparison, and these are the examples that I use as contrast, okay? This is an extreme contrast, but that's where you start to look at, the narrow forming borders, the no basal margin, and whereas a very strong basal margin to the orange, and the thicker borders on the forewing. It's like these things don't work 100%, and they're only kind of like guides. And generally speaking, if you're in the habitat, like if you can look over and see a garbage dump, or you can see an old couch or a car, then it's probably an acorn. I should put that in a key. And then there's an animal. Now this is something that's happened relatively recently, but it's of interest anyway. Anna's blue is a butterfly found in the Cascade Mountains and the Sierra Nevada mountains of California. It doesn't penetrate very far into British Columbia. And where it does, where it gets farthest north is on Vancouver Island, not necessarily in the Cascades. Now what I'm going to go through is we're going to talk about the dorsal aspect a little bit, the ventral aspect, at least to the extent that we discuss these spots. And one of the things you'll notice right away is it's got orange on the forewing, whereas Acmon never has orange on the forewing. So right away, if you see a butterfly that's got orange like that on the forewing, you eliminate Acmon and Lupin Blues, and you get into this group. I see this expression of metallic blue scaling there. That's kind of a cool thing, and especially in live photographs, or if you're there in person, it's even better. And then the extent of the orange development, that's going to be serious over the next 2 species, or 3 species, including this one. And so, we're talking about it. See, this is the same taxon. In fact, probably you could have collected these things on the same day, and it's like total suppression of all that stuff we were just looking at, right? I mean, these are the same butterfly, okay? And that's just what you learn to expect. And it's really obvious to anyone who's been on like one of our field trips into these areas where, well, I think I see an Anna's blue. It's like, I think it's an Anna's blue because it's not water walls or anything else. This one doesn't have any spots. I don't know what to do. And that's pretty characteristic of this taxon. Now, the other thing is in the females, this degree of the orange margins that you see. Another name for one of the members of the group is orange margin blue. Well, this would have to be called, if you're going to use the same, precedent, the partly orange margin blue, or the, maybe somewhat orange margin blue, almost orange margin blue. And again, the array of spots on the ventral surface of the females are basically in accordance with what we saw with that well-marked male. And another example, and this is where the markings are even more suppressed. But again, now the reason why I use this one, it's not the grandest specimen, but you can see the barest indication of these little marks. There's a little bit of silver there, a little bit of orange cap. You can kind of see where the orange band here has faded to the orange band there. And this is just part of what goes on in life.
What's this?
Annas.
Annas.
Okay, and then these are some natural photographs, and again, you can see that little orange there? I mean, there's where it ought to be, and that's a dead giveaway because it doesn't have tail. This one is, actually more typical because it has a little more, but it's not really boldly marked, okay? And then this is, again, a female, which is pretty boldly marked, but we get both kinds. Now, the other thing that's important is, I picked this one because it shows pretty extensive orange, but you have to call this one a pretty much orange margin blue. But this one here has this crescentic, highly crescentic shape to these margins, and we'll see in a minute when we get to the real orange margin blue that that's pretty critical for determining which species they are. And then we can come back to these if at any time you want to see them again. That's why they give me this machine, right? I'm just going through all the different. Okay, now there's a good example of, again, of how it's only partially, it's not very extensive, and you get the real presenting aspect. Oops.
Was all that Anna's?
That's all Anna's. And to be honest with you, single place in Kittitas County, you can get all of that variation in one day. So this is a northern blue. And a northern blue, you know, it occupies a very large area, and in that large area, there's also a lot of variation. But there's one thing about it, it's more consistently well marked like this, okay? By the way, there are genitalia differences that will distinguish these butterflies, but I thought, you know, since I'm going to do a little bit more genitalia later on, I just didn't want to get too prurient, you know, throughout the whole thing. You guys know what prurient means? You're supposed to laugh when they say that. All right.
But do you get excited when you see their genitalia?
That's for me to know, buddy. Sometimes it's not anything I've dissected before. Now, there's a lot of similarities between the Anna's blues and the northern blues on the dorsal side, including the suppression of the orange on the forewing. But you know, again, on the underside, they always tend to be much better marked. And also, instead of being like a chalky white or a basically pale off-white color, they have a tendency to have a fawn. I don't know what fawn is. I'm just hearing somebody, Nabokov, I think, use the word fawn to describe it. It's kind of like a tannish brown color. I mean, come on, fawn. Anybody heard that except Nabokov?
It's probably taupe, you think?
Oh, there you go. I'm sure glad I asked. All right, and of course, you know, this one belies what I just said because this is very, very pale. But again, you can see that, you know, the expression of the orange here is much more than your Anna's blue, but it's not as much as we're going to see in a second. Now, this is an image that came from Butterflies from Canada, and I use it because it gives you kind of a rundown of the basic similarity. These are actually subspecies of the northern blue that exist all the way from British Columbia to Newfoundland. And you can see that there's a consistency over that geographic range. It isn't too dramatic. Whereas with Anna's blue, you almost say, there's an inconsistency that is in fact the character of that species. You know, it's very, very inconsistent. And that's true all the way through California. And one of the things that I want to talk about before we go here is that we talked about Anna's blues being kind of like not especially well marked or less well marked than the rest of these butterflies. The butterfly we're about to look at is especially well marked. Now, all of these things, in fact, maybe you didn't know this, but almost all of these butterflies will hybridize with one another occasionally. Hybridization is a fact of life, and really there's a lot of reasons to believe that hybridization for species that are constantly in flux, like the post-glacial dispersal populations would be, trying to adapt to novel situations and rapidly changing ecological characters. They're going to try to take advantage of whatever that they can. And if there's a genomic advantage to be taken, but it resides in another species' genome, then if hybridization takes place that allows you to, for instance, detoxify a certain substance that this other butterfly has been living off of in a certain localized plant, and you are able to get that through hybridization and introgression, F1 breeding back to either of the parent species, they're going to do that. Well, what I'm really getting at is, what happens when you have one of these very, very poorly marked taxa, like Anna's blue where the orange is really suppressed, and there's hybridization that takes place between it and Melissa. I mean, I don't know this one that is extremely well marked. I'll maybe just get to that so that you can see it.
Talking mostly fertile hybrids?
Well, F1s oftentimes are infertile between themselves, but they'll backcross at a higher degree of fertility, okay? And the degree of fertility is maybe does dictate how successful introgression is, but it's also something we have to consider that there are controller genes, I don't know what else to call them, genes that are responsible for seeing to it that developmental functions that could be upset when hybridization takes place between two distinct species like horses and mules. Horses and donkeys make a mule. Mules oftentimes don't even have genitalia. That's primarily not functional if you want to be reproductive, right? So it could well be that and we know in fact, that there are things that are in place to protect those kinds of genomic functions that are essential for continuation. In other words, the species character is protected, and there's this zone of friction that isn't going to compromise the basic integrity. Hybridization takes place maybe fairly regularly, but it does not compromise species integrity. Now, some of the mechanisms are beyond my ability to. I'm not even sure that science knows. I think that there's some regulatory things that we know, genes that what Kiyoshi talked about at one time, some things that have these super effects on a large body of genes, or maybe even things that repair damaged genes. But You know, we know that hybridization is a functionally important aspect in current butterfly interactions. But what it means in the long term to me, it seems like obvious. It's not going to exist if it hasn't been functional. And since it still exists, it's like there's got to be some selective advantage for it. I'm going to keep running through these things. Well, really, oh, there we go. Now, I mean, come on, that's an orange margin blue. And you notice that, you know, there's really not a concentric shape to these margins here. It's pretty just solid, okay? Now, if you have one of these poorly marked ana populations, and there is hybridization taking place, this thing here, which is actually exceedingly well marked, is not going to show the effects of what, you know, a poorly marked influx in that gene pool could, you know, reflect. It's going to maybe show some reduction of pattern or whatever, you know. But ana is going to show, wow, this is big orange. So like where there's hybridization going on, a lot of times a lot of people have been confused about what's actually happening. What I want to reiterate, and I will do it now and again, is that these wings here, these are, you know, billboards. They're billboards for deception. You cannot take from looking at that wing, if there's any single aspect of that, it might be important for mate selection and certain aspects of it, like these metallic spots, but you know, like where that spot is with respect to that one, or you know, where this spot is with respect to this one over here, you know, butterflies aren't saying, wow, oh, what a babe, look at that spot, you know, it's like, none of that's important to them. They're not going to, you know, make those kinds of pattern discriminations for the most part. There could be elements within the wing pattern that are important. So, if that's true, then all the markings that we use as human beings, like, okay, this is has a spot there, this got metallic on the edge, this is orange. I mean, we're really kind of hoping that we're getting things right and it helps us identify species. It's not necessarily important to the taxon, so we should probably take our own advice on this matter, all right? Now, the other thing, too, is seasonal forms, which, you know, you don't see many blues. Wow, it's pretty nice picture.
What's this one?
Melissa's.
What's this now?
The seasonal variation, you know, we know veined whites, you know, in the spring they're very, very well marked, and in the summer they're virtually not marked at all. And that's just a simple matter of phenological difference. It has no real genetic basis. Now, this is, you know, what I was talking about before, and this really does help a lot. If you're in a place and you're wondering if what you're seeing is Melissa's or Northern Blue, this is really good. Almost always works too. Suppression of the forewing marginal band and the concentric shape of the one on the hind wing. Even if it's extensive, it's still going to be some aspect of the, you know, the same difference you're seeing here. All right. And yeah, pretty nice butterflies, actually. Like it. Okay, here we go on genitalia. So I didn't want to bombard you with genitalia, but now people ask me all the time, you know, how important are genitalia? Why do we have to know about them? Well, here's the deal, right? On the left there, you see this is the Batoides group, buckwheat blue genitalia. And on the right is the Anoptes group, buckwheat blue genitalia. You can see they're dramatically different. It's almost like, wow, man, do they belong in the same genus? Well, we actually have seen species in Central Asia that show the evolution from this type to this type, and they actually are teleologically related to one another. And actually, if you get to be really familiar with these butterflies, and in any way seen blues, you know, so they have this fringe around the ventral part of the abdomen where the genitalia exert. You can actually, with a hand lens, go in and see with the hand lens without actually damaging the butterfly. But you have to be pretty good. You can tell the differences between these two. I don't really recommend that for, I mean, like, I don't think Jeffrey Glassberg would be real happy if we said, yes, with a hand lens and tweezers, you too can tell genitalia. That's not going to be right. Well,
Tell us which of the void parts and girl parts you give.
they're all void parts.
All void parts,
Yeah.
thank you.
These are just the valves. I excluded everything else. There's nothing useful, and well, you can see this is the other part. And most of what you see as differences are a matter of how they're turned. The only real useful differences are between these valves. All right. Now this is where it gets really tricky. This is a square spotted blue again. I'm just reviewing the genital conformation. And then here we have a butterfly that I have identified as the Heracleoides feeder. And everybody's saying, oh, that's cool. I mean, how many people can say Heracleoides? There you go. All right, now this is, I mean, it doesn't, currently it's not named. It's going to be named very soon by Steve Kohler and Andy Warren. And they were pretty sure, this is one of my new names, by the way, Heracleoides blue. I'm almost certainly you can come up with a better one. Or someone already did, Hercules-like blue. That sucks too. Well, you guys think about it, maybe you can come up with, this is the blue that a lot of us see in eastern Washington on mudboats. Obviously, it's one of the things that you can see almost anywhere in the montane region. What you do is you learn the buckwheat, which we'll see in a minute. Once you see this buckwheat, you'll be driving down and you'll see a little patch of it, and you'll stop and you'll go over there and you'll have this blue on it. That's almost a sure thing. It really is kind of amazing. On a field trip I went to, when was it, 5, 6 years ago to the Blue Mountains? We did that a couple of times, and everywhere we found the buckwheat, we stopped and found the butterflies on them, and that's how closely associated they are, okay? And there's some variation, there's no doubt about it. The butterflies are not, consistently one way or the other, but there's basically an aspect to them that really distinguishes them from their nearest relative, or the one that's most often in company with them, one of the dotted blue complex, which is, instead of being, like a pale tan or even whitish, tends to be more of a cream and have, maybe the spots more separated, whereas these occur in a band. I don't find anything in any of that stuff that people tell me to be so completely reliable that I want to repeat it, because, people say one thing, somebody said, well, Pelham said, and then somebody else says, yeah, well, I heard, and then the next thing you know, it's like there's this established rumor. And I don't know. Fritz, I've heard a lot of stuff about Fritz. People say, well, this spot, if it's triangular shaped and it has a brown, if it's convex and it has a, I just don't find that stuff to be really useful. It hasn't worked for me, and I've been doing this for 40 years. Sometimes I say, people say, how do you know what it is? I says, I've been looking at them a long time, and that's about the only thing I can tell. This is a pretty standard example. Notice this black line. I didn't include Dave's images. I should have, because this is a perfect example of the Heracleoides feeder, as opposed to the other one, the Glaucon umulatum feeder. But
Genus again, I'm sorry.
Euphalotes.
Euphalotes.
Would you get on the same page, man? I've been working on you for a long time. You even got the sheet of paper right in front of me. Yeah. Now this is a common sight also, and again, I would almost say that this whitish color almost has a blue aspect to it, which is, sort of like what you might see in the fields, like a bluish white color versus a brownish, which is the other butterfly that's often going to be seen with them, which we'll get to. These are tough guys, I'm telling you, they're just tough, and a lot of times you have to become more familiar with the plants than you are with the butterflies. And this is Heracleoides, Eriogonum Heracleoides. Hercules, Hercules buckwheat. Combinations kill me with butterflies and just doing me in with plants. Then there's a thing about this buckwheat where there's this leaf, petiole, or what do you call that? The leaf that's halfway up the flower stem. It forms like a little elevator. It's more obvious. There you go, right there. See, there's a bract.
Yeah, they're called bracts.
Yeah, and it's very, very characteristic. Most buckwheats will have this flowering stem come straight out of this bushy plant at the base, and it won't have these bracts in the middle of the flowering stem. So like when you see these, you can be sure it's Heracleoides. Unfortunately, not every Heracleoides has these bracts, and so
Rings of small leaves that they're technically called bracts underneath the flowers.
there you go. And
But the converse is reliable, is it, John? When you
Yeah.
see the bracts, you know it's...
yeah, the only exception, no, actually there is one exception, but it's such a wacko look as it gets. Eriogonum thymoides. That's the one up pincushion thing, and it has very small bracts. Of course, the little things won't be big off the ground. Now, you guys, how many of you have seen our newsletter? All right, what's it called? Why is it called Gnome?
Eriogonum.
There you go. I was accused of doing too much Gnome. We were walking along Snowy Coulee, and we have lots of Gnomes, and I mean, there's probably 7 species there, and I was, talking about it as I was wont to do. They were, we're pretty cool, because some of them were blooming, and some of them were just coming up, and I was pointing out, well, this is this Gnome, and that's this Gnome. It was Lindstrom, actually, I think it's probably what's all this gonna about? You know, it's like, and that, it's an appropriate title for the newsletter, actually, a prologue. I'm gonna go through a couple more images of the Heraclea. Oh, that shows the bracts again. Great.
Actually, rubber ring around the whole.
Yeah, no, that's true. But it often occurs in these kinds of situations. Now, this is buckwheat blue heaven. I'm serious. You know, well, that's butterfly heaven, but I mean, there's lots and lots. You just walk through these and you'll see the males perched on the flower heads, and every female that shows up, they go merrily dancing after her. This is a great place to look for these butterflies. It's a good place to become familiar with those buckwheat blues before you know, like try to identify them at a mud puddle, because you know what you're dealing with when they're sitting here. Now,
John, what sort of elevation are we at? A couple thousand feet?
Yeah, it goes right down, actually, penetrates into the upper edge of the steppe, especially along ridges and in situations where, you know, maybe there's a ponderosa pine and a riparian association, and then turns into avalanche crab apple, etc. And you'll find, you know, the thing extending pretty far into the steppe. Now, this is the one exception to the Heracleoides feeding. This is Eriogonum douglasii, and it's actually kind of related. And we've now found like probably 8 colonies that use this plant instead of Heracleoides. Of course, we were all excited. Wow, a new sibling species! You think it's nothing like that. It's just, you know, it translated to this plant. But that is something that you can also keep an eye on. And I'm just warning you on account that you may come across, because this is a typical habitat for the douglasii. And in fact, it looks like Kittitas Valley, doesn't it? And therefore, you know, you guys can be expected to see this. If you see a blue associated with that, you know, you heard JP tell you that, hey, you know, they do use douglasii. I just found it in the colony.
Where again will be there?
Well, I don't know if that is Kittitas Valley. It does kind of look like it's dropping out of the Wenatchees into, like, you know, somewhere a little bit east of Cleon. But actually, this looks a lot like Kalaka Pass Road, the hill to the west of it, you know. And sort of, it could be a number of ridges like that. But
Oh, there's a mountain there in the right up. What's that? No, it's white there.
oh, yeah, I don't know. Of course, it's probably in Montana or something. And now this is the difference between the Heracleoides feeder. Or maybe, I don't know. Yeah, no, this is the Heracleoides feeder there, and this is the one that uses, this is the low elevation glaucon, which is out in Schnebly Coulee in April, and it's probably using Spirocephalum most often, although it might use a volifolium as well. Now, I got some pictures of the volifolium so we can maybe even see what that is. And the glaucon is much darker, blockier. You see that there's dark scales, you know, that are in between. So like the coloration, it's basically same central color, is whitish, but it's got a lot of dark scales over the top of it, so it has a duskier appearance. And also the spots are blockier and thicker. So the whole butterfly has, you know, a blockier aspect to it. And anybody know what that is? Come on, it's a checker spot. Good, good. Not even the right genus. Come on. I'm not going to have to go to Dave Nunley in mine. All right, this is Clausini Acastus steropi. This is the normal form of the male. Females are black, dimorphic, really. This is probably, you know, in a place like Montana Creek or something
Oh,
like
I know
that.
that one.
Huh? Well, you know, this is all iffy. It's, you know, like, we just not know. We don't know. But yeah, the last conversation that I had with Andy Warren on the subject is like, hey, we'll go with that. Now, this is the buckwheat that is using primarily, is Spirocephalum. And this is a buckwheat that really does prospers in really dry steppe habitats, and really doesn't look like a buckwheat until it blooms. And in fact, I have to admit that I spent probably the first dozen years of my life wandering all through these plants without realizing that they were buckwheats until, you know, they bloomed. Hey, you know what? Buckwheat blues don't give you the blues, you know. So my buddy was halfway right. You know, I should have regaled. No, I wouldn't. I'd be living it down. Okay, now this is ovalifolia. Now, I want to tell you, I've never really seen anything quite like that out in the desert country. I've seen something like that at higher elevations. But Dave, have you ever seen anything like that?
Only at the high elevation.
Yeah, and at high elevations, these leaves basically are much smaller too.
Yeah, very
Right. So this is another potential food plant. Now, the interesting thing about these glaucon low elevation populations is that they're almost unique in the Northwest. In fact, Andy didn't believe it until he saw it himself. Almost unique in that the adults emerge before there's an available nectar source. I mean, these things are almost always associated with their food plants, both as nectar and for the adults and food plants for the larvae. So, however they're sequenced, they managed to get around this somehow. I don't know. I mean, a lot of times I've got records after record from like Schnebly of these butterflies flying and there's no buckwheat blooming. Not even the compositor that the Sheraton lay their eggs on, they're not even close to blooming by the time that these things the adults are flying around. So that's very unusual. Almost every other buckwheat blue that you're going to encounter anywhere is going to be associated with, either you're going to see it on bud, which doesn't really count, or you're going to see it associated with its food plant, highly associated with its food plant.
So both as a nectar plant and the host.
Right. Now, they will nectar on other plants, don't get me wrong. They will nectar on other plants, but they definitely have this real close association. Now, I'm going to be doing a presentation on the way for Heidi to ask me. She's not here now, so I'm not giving her a hint. But I've got two more. One's on butterfly classification, and one's on basic butterfly biology, in which we can get into the issue of how important a food plant is to a small butterfly. So a food plant is like a mountain to a small butterfly. So its presence in its environment as a source of sustenance is really important. This presence in the environment as both as a larva and as an adult is important. And its presence in the environment as a geographical location where they can sit on this plant and locate females. Of course, females are coming to the food plant. They've all emerged in the general vicinity. So, that's really important. The buckwheats are really important for buckwheat blue biology, and you can see that yourself. You go out and you watch how they relate. Okay, this is another example from Oregon of the same creature, the glaucon, from out in dry eastern Oregon. Again, I mean, really pretty easy to tell apart once you get used to this whole duskiness aspect to it, this heavy black line, which is interesting because the high elevation ones still have that line too, so they're not nearly quite so dusty. And let's see, I'm not exactly sure what that is. You know what it is? Buckwheat?
I'm way out behind this.
Yeah, okay. Oh, there, undulatum, okay. Now, what we're looking at is the next sequence of creatures, and that's the high elevation glaucons. Now, we don't really know that they're unrelated because anybody who's gone to Schnebly and the Chumstick Mountain will know, that there's this latitudinal or elevational gradient of it was what, 2,000 meters, something like that. And maybe not that much, but I mean, it's significant. And you have an alpine, subalpine habitat on top of Chumstick Mountain. You've got the steppe habitat in Schnebly, and yet there's still green hair streaks. The Sheraton's green hair streaks in the same places. So this might be a similar kind of thing. You've got glaucon blues up on Chumstick Mountain, and you got glaucon blues down in Schnebly. They may actually represent the same thing, but representing an adaptational preference instead of a specific or subspecific difference. But this is an umbulatum, and a very, very characteristic plant. Really more widespread than people realize. It's just that it doesn't exist like a lot of buckwheats do in these large-scale landscape mosaics where you walk into a habitat and pick out the number of the buckwheat species, just visually. In 15 meters away from you, you can count 20, 30 plants. These things are closer to the ground and a little more obscure. That's two different varieties. One is the lower elevation variety. This one, and the other is the more typical high elevation variety, which has the leaves pressed to the ground. But the color, that's the color. They're dark green and they've got a pale. I think all of them have villous underside, don't they? Yeah.
Figuratively, not almost all.
Yeah. Come on, dude. There we go. And these are more examples of those. Again, this is the higher elevation form. So you can see that it's really not quite as dusky. I don't really, understand all that, but it's still got that black line along the edge. It's pretty characteristic. It does kind of distinguish them from, And you're not going to see a lot of female buckwheat blues with blue scaling like that, but you're going to see a few of them. And that's only if they perch for you with their wings spread, which is not usual for any blue. Okay. Now we get to, I say, remember I talked about simple at the beginning, you know, the arrowhead blues, silvery blues, you know, the pygmy blues, and then all the difficult. This is a headache. This has gotten worse, and I haven't even told Dave, this has gotten worse just in the last 2 months. So, give it a shot, okay? This has got the genitalia type, and it turns out actually that this is really interesting because Steve Kohler from Montana found an entirely new species, and it has a dramatically different genitalia than anything else so far described. And there's at least 3 other close species in the buckwheat blues that don't occur in the Northwest, or not until now. And it's being described, and it's really interesting in as much contrast with this, but it's not really towards any other direction. It's not like it's halfway between one thing, this, and another. It's kind of like this, but in a direction towards something totally wicked. Now, this is the real Anoptes. This is Euphalotes Anoptes. This is the dotted blue. And this is amazing, or spotted blue, for bird. This is an amazing distribution. It's all the way from the southern Sierras, all the way through Oregon, all the way into south central Washington. And the amazing thing is that I've had a chance now to look at these populations. I've seen a lot of them from the south, from Tuolumne, you know, in and around areas of Yosemite. Man, they all look about the same. You know, it's like, wow, that's striking for a butterfly that has the potential to really be pretty dramatically different, especially in a range that large. But, you know, they really kind of hang together, you know. They're kind of a generic, you know, buckwheat blue to start with. But the cool thing about these things is that they feed on this sorry little plant, Eriogonum nudum. And I mean, I'm not kidding. When I say sorry little plant, there's some leaves right there. There's 1, there's 2, maybe 3, 4. Then there's a stalk and there's these little flowers up here. Now, you think, well, that's not much of a plant to start with. And it's not like they cover the ground like a carpet either. But on top of that, throughout most of the range of the spotted blue, throughout most of the range, it's sharing this plant with Gorgon copper and an Acmonoid lupini blue. Okay, I'm a member of that group. The lupini and the Gorgons feed on the leaves, such as they are, and the spotted blues feed on the flowers. So at least there's some partitioning of their resource. You begin to wonder how there's any of these things left anywhere with that kind of predation level. I mean, I got another picture. Oh, that's much healthier, right? You get an idea. Yeah. It's kind of a sorry little plant. And, you know, I honestly hadn't seen any in my life, except for pictures when I went up with Dave and he showed me this. And I'm walking along looking at and seeing these blues, you know, and I'm thinking, what a way to make a living, you know. Wow, this is stretching the ability of that plant. But it's even more amazing when you consider that this same association exists all the way through this huge geographic range. It's like, man, there must be something going on that's really beneficial. It must not be too bad for the plant either, but really beneficial for the butterflies that decide to use this. Now, in Washington, this was only recently discovered to exist. And the interesting thing about it is it's only about 500 meters above well-known colonies. And we're discovering other specimens of the thing now in collections at the National History Museum and various other collections that people have donated. I've even found one in my own collection that was misidentified by me. I mean, we get an unidentified Anoptes group blue from 4,200 feet. You don't know anything about it. It was given to you, and it's easy to make a mistake of lumping it in with the lower elevation things. But how extensive it is, we don't really know. Bob Pyle, bless his heart, and he's our own traveling gnome. He promises to come back and give us a rundown on his big year, and only after I get done looking at everything that he sampled, so I can identify. But he discovered this thing, and now we know it's more widely spread. So it's a new butterfly for our state. And it's a new butterfly for our state because the one that we thought was the traditional dotted blue. This is the other name I made up. You guys are going to have to pass judgment on that. Colombian blue. I mean, you can see how I got to that way. 3 or 4 years ago, this wasn't, well, let's make it 5 years ago, this wasn't a butterfly we recognized as a distinct species. I misspelled this poor guy's name, added an L to it. That's the guy that helped me publish my catalog, so don't anybody tell him. He's severely offended. This is the butterfly that you're going to see. This is the Anoptes group butterfly you're going to see all along the east slope of the Cascades. It feeds on this wonderful plant that's just really, we'll look at it in just a second. This is the one we see all the time, and it's circumscribed around that plant very tightly, except, and I'll get to the except in a minute. I'm going to kind of go through to what they look like. You can see they're not radically different from the Anoptes. If you have enough of them sitting side by side and you look at them up and down, you say, oh yeah, I can see some differences. You need a little more dusky, a little bit bigger line here, maybe something. But, it's not like they're like, whoa, these are really different. It's just that they're biologically completely distinct. This is the plant they use. This is Eriogonum composite. Now, you got this huge array of leaves here. You got these really healthy flowers, these things are just juicy to the max. There's at least 6 Lycaena species that use this plant at some stage or another. Coppers, blues, and hair streaks. So this is definitely a gill builder, and it's definitely one of those plants that if you live in a hostile environment, you want to ride that horse as long as you can, all right? And you can go a lot of places to see, this kind of an array in eastern Washington. A lot of you guys have. You've seen this plant widely. I talk about it all the time. Same thing, Oregon, wherever it occurs, you've got this Columbian blue associated with it. And it was like, that was our first clue to it. Now, it's not restricted to this, as we'll get to. It's kind of an interesting thing, but it is so commonly associated with this, so widely associated with this, that if you're climbing up and down the hillside and you see this plant, it's easy to recognize too. It's got these big heart-shaped leaves, and it's got, of course, this very robust flower stalk. When you see one, it's like, oh yeah, that's the one that JP was showing us, and that's exactly right. And you see the little blues sitting on it, and you're like, hey, there's the Columbian blue. Now, I'm just going to go through some more images of what the adults look like, and of course, wild images are always better. Now we get to the and or if maybe. Okay, now this is a plant. Kind of looks like Eriogonum nudum that the poor little Anoptes have been defoliating all over. These are not. These are elatum. They're a much more vigorous and robust plant. There's a better idea. You can see, you guys probably recognize this, like this pinkish flower that kind of goes on. There's places in eastern Washington, as you look over, like this looks like, like right below, come on, Reese Creek.
Our house.
Huh?
Our house.
Oh, your house, exactly. In the summertime when these things are in bloom and you're standing, you look out, it's like this sort of pink haze that goes on forever. It's just, now, we first discovered, a long time, it was me that discovered it because there wasn't really anybody around to talk to about it except me. And so I was talking to myself and I said, hey, there's a butterfly, a dotted blue that uses this plant. And that's like, okay, so it's a butterfly that uses 2 food plants. But the thing is that the composite, the buckwheat that the other butterfly uses predominantly is an early summer or maybe even late spring blooming buckwheat that exists in more montane habitats. And this is a late summer buckwheat that exists in more open habitats. A perfect example of the kind of place it occurs, as you can see. It's away from the trees and then extends well out into the steppe. And because of these flight period differences and because of the elevational differences, it was like, well, maybe we're sitting on sibling species. Well, we had a young fellow, Merrill Peterson, that did a lot of his doctoral research was on Euphalotes genus, especially landscape mosaics. And he did some electrophoretic work on it that indicate, well, the electrophoresis basically can show you allelic differences between populations. And so the idea is if you have 20 villages in Bavaria and you measured all of their blood types, you would see certain kinds of concordances associated with how much discourse they had with each other. There's not going to be, things that are right adjacent to one another that have complete discord. There's not much intercourse going on between them, right? And so, if you see uniformity, then you think, okay, well, the reason why these, the O blood types and the B blood types and the AB blood types are relatively similar to one another is because there's interbreeding taking place. I just got last week the bad news on that. One of our new authors on the Butterflies of America website, he said, it turns out that some of these alleles are multivalent, and they may actually have the same
weight. Now, electrophoresis works by driving these proteins in a gel, and one goes farther, one goes less far, because they have a different molecular weight. And when you actually show that that's not so, then the results of an electrophoretic study are useless. So it turns out we may have actually sibling species here. You sure can't tell by looking at them, though, and I hate to have to make a living off of, I have that one, that usually, maybe not going to be a lot of them, and that one usually come positive, but that's where we're at with this right now. We're looking hard. There's definitely no doubt about it. They fly a lot of times in the same areas. You go in spring, you get the one set on composite them, you go in late summer, you get these things will actually fly, the blues themselves will fly late into August. And long after the composite them are, they're already crispy toast. And these plants, now you think about it, what an advantage it is to be able to have both strategies. You were thinking there's this butterfly that had these two strategies that wasn't being compromised by gene flow. And if it turns out that in fact there is no gene flow, then that model doesn't work. But anyway, it's pretty obvious where one came from. One is utilizing these montane habitats with the composite as a food plant. The other one is now enabled to move into steppe. This is also one of the few buckwheat that readily spreads into disturbed habitats. It goes into these habitats at will. I mean, you guys got them all over your backyard, right?
Yeah.
And probably see them every roadside in and around.
Yeah, well, if it's too disturbed, no.
Well, yeah, I mean, if the graders were just there, give them about a decade, though, they'll come back, right? Have you guys seen any of the blues on them?
Not so much. I don't see it that thick.
That's unusual to be that big.
Dave,
Sorry?
you seen it that thick?
I'm sorry?
Have you seen the lot of that thick?
Absolutely.
Absolutely. That's my experiences. Now, the other thing that's cool about this, and I'll probably, let's see, I want to make sure that, yeah, I want to go one more, I think. The other thing that's cool about this, the buckwheat blues, maybe I didn't make this clear, they don't eat leaves at all, they only eat the blossoms. So we look at the composite plants, right? They have big, flower heads, and it's like a buckwheat blue can sit in that flower head, eat all the flowers at once, all the seeds as it gets ready to, go to seed, and it'll be done, and then it'll drop down and make a pupa, right? These flower heads are much smaller. And so like any blue butterfly that's going to be, using this as a food plant has to eat one, and then it has to crawl down the stem and go up to the next one, which is all risky business, okay? And a caterpillar, of course they do come with an army of ants. They don't eat leaves at all, they only eat blossoms. So we look at the composite plants. So I'm basically done now. I don't know what time, did I run over? I'm done, and if you got any more questions or any place you think that we ought to go, anything that you think I was vague upon, or anything that I could correct or rectify? Come on. Yeah.
I just want to add something.
Okay.
The way you find these caterpillars is looking for the ants.
Well, that helps. You can look for them without looking for the ants, but then don't have to know exactly where to look. But the ants, a lot of times
One
when
of
you
the
look
best places to
yes.
spot them is to look
Yeah,
for the ants.
absolutely. Mm-hmm.
In the last couple of issues of the National Butterfly Association, there's a couple of articles on buckwheat blues.
Yeah.
Two questions. What do you think of it? And I think you've already answered that.
Yeah.
And then the other is there was a statement in there that just blew me away. There was something about in the state of California there are 100 different species of Ariogeny.
Yeah.
Is it that?
Yeah, well, down there, yeah. See, what happens down there is that you've got this expansion, contraction, differentiation, isolation, speciation without extension. So, in a place where, you have, that's why the tropics have so many more species than oil. The rate of speciation is the same across the face of the planet. It's just that everything gets wiped out further north. In the south, you get compacted. And so, you add species each generation. There's some extinction, but I mean, you're able to stockpile things. Yeah, there's a lot of buckwheat species down there. But that article in the NABA journals by a guy named Gordon Pratt, and I get along with him really well. Andy Warren and he are at war with each other over this. He is talking about these things from a perspective that's California, right? Now, when was the last time the glaciers came and wiped out anything in California? See, that's the problem. He has this perspective of the world. It's way different. And that's another thing that's really important. I just want to do basic butterfly biology again, because You know, it's really important for us in the Northwest to recognize the significance of the climatic revolutions that have taken place here. And to recognize that every butterfly is like, it's on the edge of trying to get by. You know, right now we're seeing like the, kind of like the edge of the last, you know, expansion. But if the glaciers came again, it would start crunching everything we had in south. Where do you think Lorquin's admiral came from? If you look at the total distribution of that butterfly, it's California. You know, it got here kind of recently. It's penetrated into BC, but you can talk about that in terms of, you know, tens of thousands of years or less. It's just recent. Certainly the California sister, or, you know, the Adelpha californica, that's even more recent, and it hasn't gone as far. It's just barely getting into Washington. You can look at everything in terms of like, we have this thing going on, but it wasn't so long that it wasn't going on at all, because we were under a mile of ice. Gordon Pratt, you know, is a wonderful researcher, nice guy, but he has no clue. And he should have been writing about our buckwheat blooms, especially when we both, Andy and I, both wrote to him to tell him about it. He doesn't even know about these Heracleoides feeders. Why? I don't know. Everyone else does, you know. So yeah, unfortunately, on the other hand, it was good, because that was one of the technical articles in American Butterflies. It wasn't bad for you know, the general audience. You know, so I like seeing that in America. I'm probably going to, you know, actually get this journal.
Great.
Yeah, absolutely. Absolutely. Any more? There's another question hanging there, David.
Just that you mentioned a black margin on the Heracleoides feeder,
Uh-huh.
but I saw the black margin in the other news as well.
Well, in the Anoptes.
Yeah.
Yeah, it will show up in the Anoptes, but the thing it's contrasting with the, it's not in the Heracleoides feeder, it's with the other, the Spirocephalum and Undulatum feeders. Is it? No, it's the other way around. It is the Heracleoides feeders. The other one's the lacking plant. But again, it's not, you know, nothing's solid, you know. It's kind of like, you know, you get about 20 photographs and you're maybe 2/3 of the way towards, you know, an initial understanding. You have 100 specimens, you know, you have to look at them all and say, well, geez, you know, just about the time I think I'm starting to understand this, you realize, well, you know what, it's still iffy. And you know, a lot of people tell you that, oh, this guy can't identify Fritz. You know what, I've decided, well, you can identify 85 to 90%, but you know what, the best, the very, very best in the world, you know, and one of those is a friend of mine, Paul Hammond. I mean, he'll pick one up and he'll say, I don't know, you know, could be Coronas, could be Zuri, stick it back there. I mean, how are we supposed to know? There's no way. You know, that butterfly probably, you know, knew what to look for. He knew what female to look for, but, you know, those wings, they're not necessarily any kind of an indication. They're just a billboard deception. And it's really important to think about, what do butterflies do better than almost any other animal? You know, they deceive us by mimicry, right? That should be our first clue. It's like, yeah, I'm looking like something else. Hey, humans, wake up! I can do these other things. I can fool you. You know, that's what they do. They fool us. And anybody who thinks that they can, you know, write a guide, you know, like I say, I should put that in the key. If you are sitting in a junkyard and you see a little balloon with orange on its hind wings, it's probably an aqua. It's not as good as these other characters sometimes. Yeah. All right, Dave.
Yeah, Stuart was asking how do you find larvae on these buckwheats. There's a real easy way to do that. Just try and cut a bunch of flower heads off and take them long enough to kind of lightly cover the bottom of the terrarium. Just let them sit for about 3 days until they dry, and the larvae will just crawl right up the size of the terrarium.
Yep. In fact, that's what Gordon Pratt does an awful lot of, and he's been very, very successful. But, you know, the interesting thing, contrast between Gordon and Andy Warren. Andy Warren is not a big life history guy. Gordon Pratt's a big life history guy. But he would go on these places and collect all these buckwheat samples, bring them home, they're all organized and everything. He's never seen the adult butterflies flying in nature. No, he has, but it's pretty rare compared to his field time. Andy's out in nature seeing all these things all the time. So it's like this yin and the yang. Together, they should hold hands and make up.
I think you need both
Yeah,
things.
absolutely.
And doing everything, one person doing everything.
You can't do it.
You can't, you gotta
Yeah.
collaborate.
Yeah, well, it's, you know, yeah, it's real important, and I definitely agree with that. Yeah.
You take all these pictures of places that seem dominated by native plants.
Uh-huh.
I don't get it. Where do you find these places? It seems like increasingly it's like dominated by aliens.
Yeah. I think probably
most of the time when you decide to take a picture, you selectively take whatever angle it is you can to avoid those alien plants. Yeah, I would agree with you, it's pretty hard to find. Harder in some places, especially like steppe habitats, to find places that don't have so many alien plants. But, you know, you do what you can to make people, you know, believe. Anything else? I'm done.